Carbohydrate is a chemical compound that contains carbon, hydrogen, and oxygen in the ratio 1:2:1. So, from the ratio of these three elements, it seems as if one carbon is associated with one water molecule or H2O. And that's exactly why the name carbohydrate, that is hydrated carbon, that is carbon which is hydrated or is in association with water molecule. The other name for carbohydrate is saccharide, which means sugar. However, there are exceptions to this introduction. What are the exceptions? So, the acetic acid, let's look at acetic acid. Acetic acid molecular formula CH3COOH. How many carbon? 2. How many hydrogen? 4. And how many oxygen? 2. Here, the ratio of carbon is to hydrogen is to oxygen is 1:2:1. That is, the molecule of acetic acid satisfies this particular criteria. However, acetic acid is not a carbohydrate. Similarly, deoxyribose. Deoxyribose has 5 carbon atoms, 10 hydrogen atoms, and 4 oxygen atoms in its molecule. Here, the ratio of carbon is to hydrogen is to oxygen is not in the ratio of 1:2:1. However, it is a carbohydrate. That means this particular definition of carbohydrate, that is carbohydrate being a chemical compound that contains carbon, hydrogen, and oxygen in the ratio 1:2:1, is not enough because it contains a lot of exceptions and contradiction. According to this, the empirical formula of carbohydrate is CnH2nON. Since there are exceptions to this particular definition, we have been introduced with a better definition, and that is, so carbohydrate is defined as polyhydroxy, aldehyde, or ketone. For example, glucose. Glucose has 6 atoms of carbon. 1, 2, 3, 4, 5, 6. Here's the aldehyde group, hydroxyl group, hydroxyl group, hydroxyl group, hydroxyl group, and another hydroxyl group. As you can see, one molecule of glucose contains 1, 2, 3, 4, 5 hydroxyl groups. Basically, more than one hydroxyl group. And it is in fact an aldehyde. Since it is a polyhydroxy aldehyde, it's a carbohydrate. Similarly, there's another part of this definition. Those compounds that give such derivative, by such derivative it means polyhydroxyaldehyde or ketone, upon hydrolysis. Hydrolysis refers to breakdown of molecule by the addition of water. So such compounds that give polyhydroxyaldehyde or ketone upon hydrolysis are also carbohydrates. Example, maltose. So the molecular structure of maltose looks something like this. So here's one part which is connected with another part. This particular molecular structure does not show any aldehyde or ketone group. However, upon hydrolysis or addition of water, one maltose molecule gives 2 glucose molecules. And what does glucose molecule look like? It looks something like this. That is, glucose is a polyhydroxyaldehyde. Since maltose upon hydrolysis gives polyhydroxyaldehyde, or maltose is a compound that gives polyhydroxyaldehyde upon hydrolysis, it's a carbohydrate. Now, this is the actual definition of carbohydrate. So what's a carbohydrate? Carbohydrate is a polyhydroxyaldehyde or ketone or such a compound that gives polyhydroxy aldehyde or ketone upon hydrolysis. Fine. Now let's look at the various functions of carbohydrates, and there are many functions. However, I'm just going to mention few. The rest you'll come to know soon enough when you study the various types of carbohydrate. First, it is the source of energy. Obviously, for example, there's your glucose. Glucose. It is a respiratory substrate. That is, glucose goes through the process of respiration or oxidation and gives waste products, obviously carbon dioxide and water, along with which we get energy. And this energy is either used by the body or stored in the form of ATP. Another function. It also forms this, not just the source of energy, but the storage form of energy as well. So storage of energy. For example, glycogen stores energy in animals, and starch stores energy in plants. Then function number 3. The plant cell wall contains cellulose, hemicellulose, and what do they help? They help in providing structure or foundation to the wall of the plant, to the plant cell wall. Not only that, the exoskeleton of insect, which is there for protection, is made up of chitin. And what's chitin? That's a carbohydrate as well. Function number 4, extremely important. The genetic materials, RNA and DNA, they contain carbohydrate as its main component. RNA contains ribose sugar. And what's ribose sugar? It's a carbohydrate. Similarly, DNA contains deoxyribose sugar. What's deoxyribose sugar? It's a carbohydrate as well. Number 5. Further, within this chapter, you are going to learn about GAGs and glucuronic acid. Both GAG and glucuronic acids, they have their own therapeutic and clinical functions, which you will come to know soon enough. So this is the definition and the various functions of carbohydrate. Now let's look at the various types of carbohydrates. Depending upon the number of sugar units, carbohydrates is classified into 4 categories. There's monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides contains the part mono, which means one, and saccharide, which means sugar. So basically, monosaccharide contains 1 sugar unit. Similarly, disaccharide contains di, which means 2, and saccharide, which means sugar. So disaccharide is a carbohydrate which contains 2 sugar units. Similarly, oligo means few and saccharide obviously means sugar. So it contains few sugar units. That is around 3 to 10 sugar units. Similarly, poly means many and saccharide means sugar. So polysaccharide is such a carbohydrate which contains many sugar units, more than 10 sugar units. Now, since monosaccharide only contains 1 single sugar unit, it can't be further hydrolyzed into even smaller units. So, monosaccharides cannot be further hydrolyzed into smaller units. Among the various types of carbohydrates that are present based on the number of sugar units, let's first deal with monosaccharide. So number A, monosaccharide. It's such a carbohydrate that contains only 1 sugar unit. That's why monosaccharide. Now, monosaccharide itself is further divided into various categories under various bases. First basis, on the basis of number of carbon atoms. So on the basis of number of carbon atoms that is present in a monosaccharide, it's divided into various types. There's your number one, triose sugar, which contains 3 carbon in 1 sugar unit. Tetrose sugar, which contains 4 carbons in the sugar unit. Third is pentose sugar, which contains 5 carbon in a single monosaccharide unit. Then there's your hexose sugar, which contains 6 carbon. And finally, heptose sugar, which contains 7 carbon. On the basis of the number of carbon, we can also predict its empirical formula, because we know that the empirical formula of a mono in general is CnH2nOn. So for a triose monosaccharide, it will be C3H6O3. Tetrose monosaccharide, C4H8O4. Pentose monosaccharide, C5H10O5. Hexose monosaccharide, C6H12O6. Heptose monosaccharide, C7H14O7. This is on the basis of number of carbon atoms. The second basis is on the basis of functional group that's present. Because we know that a carbohydrate is a polyhydroxy aldehyde or ketone, or such a compound which gives polyhydroxy aldehyde or ketone upon hydrolysis. Since monosaccharide can't be further hydrolyzed because it is the smallest form of carbohydrate, it contains only 1 sugar unit, then monosaccharide itself is either a polyhydroxy aldehyde or a polyhydroxy ketone. So on the basis of functional group, monosaccharide is either an aldehyde, then it would be called an aldose monosaccharide, or a ketone, then it would be called a ketose monosaccharide. Let's look at the examples of monosaccharide based on the number of carbon as well as based on the presence of which type of functional group, aldehyde or ketone. So triose sugar, we have glyceraldehyde, and ketone dihydroxyacetone. When glucose splits during the process of glycolysis, during splitting stage of the glycolysis, it gives 2 triose sugar. One is aldehyde and the other is ketone. So glyceraldehyde is the aldehyde or aldose triose sugar, and dihydroxyacetone is the ketose triose monosaccharide. Similarly, for tetrose, we have erythrose under aldose sugar and erythrulose under ketone. For pentose, ribose and ribulose. For hexose, we know very well, glucose, which contains aldehyde group, and fructose, which contains ketone group. And for heptose, we have glucoheptose and pseudoheptose. The second type of carbohydrate that we have to deal with is disaccharide. It is called so because it contains 2 sugar units. How exactly is disaccharide formed? Or in fact, how exactly is oligosaccharide formed? When one monosaccharide combines with another monosaccharide by forming a bond called glycosidic bond, it forms disaccharide. When at the same time, around 3 to 10 monosaccharide units are involved in the formation of glycosidic bond to form a single large carbohydrate, that's oligosaccharide. So now we know how disaccharide is formed. Now let's look at the various examples of disaccharide and its sugar units. So there's maltose. Upon hydrolysis of this disaccharide, that is breakdown of the disaccharide by the addition of water, we get glucose plus glucose. Similarly, next is lactose. The hydrolysis of lactose gives us glucose plus galactose. Then there's your sucrose. The hydrolysis of sucrose gives glucose plus fructose. So these are the three main disaccharide that you need to remember. Another thing, most abundant monosaccharides. What's the answer to this question? So the most abundant monosaccharide that is found is D-glucose. It is a hexose monosaccharide, that is, it contains 6 carbon atoms in its molecule. Okay, so now you know what disaccharide is. Similarly, let's move on to oligosaccharide. So oligosaccharide contains 3 to 10 sugar units. Let's look at few of its example. So there's your raffinose, the hydrolysis of which gives you glucose plus galactose plus fructose. So the hydrolysis of raffinose, which is an oligosaccharide, gives you 3 monosaccharide units or 3 simple sugar units. And that's exactly why raffinose is a trisaccharide. Tri stands for 3 and saccharide stands for sugar units. Since raffinose upon hydrolysis gives 3 sugar units, it's called trisaccharide, and since it contains 3 sugar units forming itself, it's an oligosaccharide. Similar to raffinose, there's your stachyose. The hydrolysis of stachyose gives you 2 galactose and then a glucose and a fructose. So how many sugar units does stachyose give upon hydrolysis? 4. So it's obviously a tetrasaccharide. Similar to stachyose, the next example is verbescose. The hydrolysis of verbescose gives 3 units of galactose. And the rest is same, glucose and fructose. So altogether, how many monosaccharide units? Altogether, 1, 2, 3, 4, 5 monosaccharide units from a single carbohydrate molecule. So it's a pentasaccharide. So tetrasaccharide, trisaccharide, pentasaccharide, up to 10 sugar units fall under oligosaccharide. After oligosaccharide is polysaccharide. It contains many sugar units, more than 10 sugar units. Now, polysaccharide, they are also called glycans, just a different name given to polysaccharide, or complex carbohydrate. I think you might have already guessed why complex carbohydrate, because it contains large number of sugar units that are arranged in a complex manner. That's exactly why. Now, polysaccharide is divided into various types itself on the basis of various criteria. So on the basis of structure, polysaccharide is either linear, that is, the sugar units are arranged in a linear fashion, or branched, that is, the sugar units that is arranged have branches. Example of linear polysaccharide is cellulose, and the example of branched polysaccharide is glycogen. Cellulose, that is a component of cell wall of plant and provides protection to plant, and glycogen, which is the stored energy in animals. Similarly, on the basis of types of sugar units present in polysaccharide, that is, whether the sugar units present are of same type or different type. So there's your homopolysaccharide or homoglycan, homopolysaccharide, I'm just gonna write till sac, and heteropolysaccharide. So homo means same, polysaccharide refers to, well, this carbohydrate right here. If the polysaccharide contains same sugar units or repeating sugar units, the same sugar units repeats itself in the polysaccharide, then that's your homopolysaccharide. However, if different types of sugar units are present in a single polysaccharide, then that's your hetero, means different polysaccharide. So in heteropolysaccharide, different sugar units are present. It's not just the same sugar unit repeating again and again. There are different types of sugar units present in heteropolysaccharide and the same type of sugar unit present in homopolysaccharide. So we are done with the various types of carbohydrate. Now we know the definition of different types of carbohydrate and their examples. Let's look at each type in even more detail. The first is monosaccharide. Under monosaccharide, the topic is structure of glucose. Glucose is a hexose monosaccharide because it contains 1, 2, 3, 4, 5, 6 carbon in its molecule. It's also an aldose monosaccharide because it contains aldehyde as the functional group. Now, glucose is present both in a straight chain form and in a cyclic form. The straight chain form and cyclic form can interconvert into one another. Let's look at both of them. The straight chain form is called Fischer projection. For the straight chain form to form cyclic form, the first carbon, which contains the functional group, it forms a bond with the fifth carbon. So the C1 carbon forms a bond with the C5 carbon, C1, C5. And the type of bond formed here is hemiacetal, which leads to the formation of cyclic form, which is also called Haworth projection. And that's all you need to know. Second is structure of fructose. Fructose is also a hexose monosaccharide because it contains 1, 2, 3, 4, 5, 6 carbon atoms in its molecule. But fructose is a ketose monosaccharide because it contains ketone as the functional group. This is the straight chain or Fischer projection, and this is the cyclic form or Haworth projection. In fructose, the second carbon forms a bond with fifth carbon. So we get C2, C5, hemiketal, ketal bond for the formation of this cyclic form right here. In case of glucose, the cyclic form is hexagonal and it contains 5 carbon atom, 1, 2, 3, 4, 5, along with 1 oxygen atom. And this type of ring is known as pyranose ring. So pyranose ring is seen in glucose. What are its characteristics? First, it contains 5 carbon atoms plus 1 oxygen atom, and it is hexagonal in structure. 1, 2, 3, 4, 5, 6. 6 sides, right? Hexagonal in structure. Similarly, the cyclic form of fructose is known as furanose. F for fructose, F for furanose. So here's the furanose ring. What are its characteristics? So it contains 1, 2, 3, 4. 4 carbon atoms in its cycle along with 1 oxygen atom, and the shape of the cycle is pentagonal. So glucose gives pyranose ring and fructose gives furanose ring. So there are various tests for monosaccharide which you can remember with the help of abbreviation MBBS. Something you might be doing right now. Even if you are not, it doesn't matter because this is important. Where M stands for Moles test, B stands for Benedict's test, another B stands for Barford's test, and S stands for Seliwanov's test. Moles test is done to detect the presence of carbohydrate. Benedict's test is done to detect reducing carbohydrate. Barford's test is done to detect monosaccharides, and Seliwanov's test is done to detect ketose sugar. See, there are various types of biomolecules present. Among them, carbohydrate is detected with the help of Moles test. Under carbohydrate, you can find reducing and non-reducing carbohydrates. So there's non-reducing carbohydrates that does not have the capacity to reduce, and reducing carbohydrates. Under carbohydrate, you can detect reducing carbohydrates from non-reducing carbohydrates with the help of Benedict's test. Again, under reducing carbohydrates, you can find disaccharides and monosaccharides. You can detect monosaccharide or test for monosaccharide from disaccharide with the help of Barford's test. Even under monosaccharide, you can find aldose monosaccharide, and ketose monosaccharide. So you can test for ketose monosaccharide from aldose monosaccharide with the help of Selivanov's test. So Mollis test helps to detect carbohydrate. Benedict's test helps to detect reducing carbohydrate. Barford's test helps to detect monosaccharide, which is reducing. Selivanov's test helps to detect ketose sugar. See, sugars are of two types. There's your reducing sugars that hold the capacity to reduce, and non-reducing sugars that does not contain the capacity to reduce. Reducing sugars contain all monosaccharides, glucose, fructose, galactose, and few disaccharides, lactose, maltose. While on the other hand, non-reducing sugars contains the disaccharide sucrose. Why? All monosaccharides, they contain either aldehyde or ketone as the functional group, and it is due to the presence of aldehyde and ketone that they possess the property of reducing others. That is, they can act as reducing agent. In case of lactose and maltose, they contain hemiacetal group, which is responsible for the process of reduction. During hydrolysis, the hemiacetal bond that is present, it splits and forms the functional group aldehyde, which further helps in the process of reduction. But sucrose does not contain hemiacetal group. It contains two acetal groups. Since it does not contain hemiacetal group, it does not contain the property of reduction. That is, it can't act as a reducing agent. You don't need to know all these in details. All you need to know is all monosaccharides are reducing sugars. Few disaccharides such as lactose and maltose are reducing sugars, but sucrose is a non-reducing sugar. The various tests that come under this topic are actually a part of your practical. But if you do want to know more about this test and probably prefer a separate video, I recommend you comment down below. I'll be watching those comments. Okay, now see, maltose and lactose, they are quite similar because both maltose and lactose, they are disaccharides, and both of them are reducing disaccharides, unlike sucrose. Maltose and lactose, both being reducing disaccharide, makes it difficult for us to distinguish between them. It is quite difficult for a person to distinguish between maltose and lactose in a laboratory. The best way is osazone formation. With the help of this particular reaction, we can easily distinguish between maltose and lactose. But before that, we need to know what exactly is osazone formation, what's the chemical reaction taking place, and what in the hell is osazone anyway. So, this is what osazone formation really is. If we react a reducing sugar includes maltose and lactose from disaccharides and all monosaccharides. If we react reducing sugar with phenyl hydrogen, what's phenyl hydrogen? So what you see right here, NH2H or NH3 is ammonia. If you replace this hydrogen with NH2 group, you get NH2, which is hydrogen. If one of the hydrogen is replaced with phenyl group, then you get phenyl hydrogen, and this right here is phenyl hydrogen. Okay, so when a reducing sugar reacts with phenyl hydrogen, it gives osazone. What's osazone? Osazone is a yellow or orange crystal. Nothing else. It's just a crystal or a crystalline structure which is either yellow or orange in color, and it's formed only when a reducing sugar is treated with phenyl hydrogen. So let's look at various types of reducing sugar and the type of osazone they give. First, let's look at the various monosaccharides, which include glucose or mannose or fructose. All these carbohydrates are monosaccharides. When these monosaccharides, they react with phenyl hydrogen, they give crystals that look like needle or needle-shaped crystals called osazone. Needle-shaped osazone. So from the type or the structure of osazone present, we can distinguish between various types of carbohydrate. If the type of osazone formed is needle-shaped, then it's obviously one of the many monosaccharides that's present. Number 2, let's take lactose. Lactose is a reducing disaccharide which upon treatment with phenyl hydrazine gives a powder puff osazone or a tennis ball osazone. Powder puff means when you dip the cotton in powder, I don't know what you call it, take some powder and put it on your face, you get puffy structures that come out, right, of the powder. Puff of powder, that's powder puff. Or tennis ball osazone, because it kind of looks like a tennis ball. Tennis ball osazone. Next, let's take maltose. Upon treatment with phenyl hydrazine, gives sunflower shaped osazone. But if we take a non-reducing sugar and then react this non-reducing sugar with phenyl hydrazine, no osazone formation takes place. For example, sucrose. Let's take sucrose. It is a disaccharide, however, it is non-reducing due to the absence of hemiacetal group. So sucrose, upon treatment with phenyl hydrazine, gives no osazone. And this is how you can distinguish between or among the various types of carbohydrates. So all monosaccharides, they are reducing sugars, and all monosaccharides upon treatment with phenyl hydrazine gives needle-shaped osazone. Number 2, among disaccharides, there are reducing disaccharides and non-reducing disaccharides. Among reducing disaccharides, there's maltose and lactose. Lactose, upon treatment with phenyl hydrazine, gives powder puff or tennis ball shaped osazone. Maltose, upon treatment with phenyl hydrazine, gives sunflower shaped osazone. And finally, for non-reducing disaccharide, you have sucrose. So non-reducing disaccharide upon treatment with phenyl hydrazine gives no osazone at all. Polysaccharides, they are divided into two types, homopolysaccharide and heteropolysaccharide, where homo refers to same. So all the sugar units that form the polysaccharide are of the same type. However, in heteropolysaccharide, where hetero refers to different types of sugar units are responsible for the formation of polysaccharide. Now, let's look at few examples of homopolysaccharides and their functions. Starch. It is the storage form of glucose in plants. In human, the storage form is glycogen. In plants, the storage form of glucose is starch. And starch contains two major parts, amylose and amylopectin. Next, cellulose. Cellulose is found in cotton fibers, jute, hemp, rayon, etc. Third, glycogen. It is the animal starch, or it is the storage form of glucose in animal. Glycogen stores glucose in liver and muscles. Chitin. Chitin is found in the cell wall of fungi as well as forming the exoskeleton of arthropods. Inulin. Inulin is very important because inulin is used in the study of glomerular filtration rate. It is one of the studies done during the test of kidney normal function to check whether your kidney is functioning normally or not. And finally, dextrin, which is a component of honey. As for heteropolysaccharides, we are going to have a different video because it contains many important carbohydrates. We need to know about the functions, types, and their structures. As for the test of polysaccharide, we have the iodine test. And that's it for the part 1 of carbohydrates.